you that it is having the pentameric structure. Again, they are linked via the disulfide linkages using another small short peptide fragment, which is called the J chain fragment. Here we can see. Okay, so the properties of this IgM is that it's the third highest serum immunoglobulin, and this is the immunoglobulin which is first made by fetus and all B cells. So all the B cells are default; they are programmed default. So default programming to produce IgM all the cases. So when there is an infection, if the infection or the antigen is not recognized properly, not processed properly, the basic immunoglobulin that a cell always try to produce to control the infection into a basal level is this IgM. So it produces IgM for the very beginning so that they can slightly go against the antigen that newly enters, and then they will modify it and produce a perfect specific immunoglobulin so that they can fight it much more efficiently. But for the beginning, they secrete this IgM. This IgM can also fix complement. This is the best complement fixing IgM, because as we can see, I have not discussed about this, that as we are having, this is called the FAB portions, right? This Y-shaped region is called the FAB portion and antibody binding portion, and what we are having two different segments. So that's why for each monomer of all these antibodies, there are two regions for binding with antigens. So that's why usually a monomer of antibody is called as, or is termed as, divalent. So it is divalent, means it can bind with two different molecules, two different antigen molecules at a time. Right now, for this IgM, if it is having all these five different monomers, and I remind you that this all these monomers are having similar heavy chain as well as they are having similar light chain. Okay, they are having similar heavy chains as well as light chains. So they are having identical 5 monomers, so they are having the valency of 5 into 2, which is giving us a 10. So they are having the valency of 10. So the 10 valency is a very very huge valency, so they can bind with that number of immunoglobulin, yeah, that number of antigens, so that they can fix the complement very well. Because the antigen for the complement fixation, the most important part is the attachment of antigen with antibody, so that we can form an antigen antibody complex, and the formation of antigen antibody complex can be achieved more quicker by this IgM, because it is having 10 regions to bind with the antigen. So it will quickly agglutinate, or it will quickly bind with all these antigens, and that's why it is quickly fix its complement. Okay, and what is complement fixation? I've already discussed in my video lectures about the antigen antibody interactions in especially in the in last kind of tests, or a complement fixation test. Okay, you can find it in my YouTube channel. Okay, and obviously it is having some kind of receptor or biological activities, like it can bind with some Fc receptor onto some surface of B cells. So that's why it is having slightly elongated polypeptide chain here. It is called also the tail piece, which is the interacting part. This is the terminal of Fc region for this IgM monomer that can sit onto the inner cell surface of B cells, which are the secretory house of this kind of antibodies. Now here we go for the IgD and IgE. IgD is slightly very common, but IgD is a kind of protein molecule which is having much more what we can say, sugars attached to it. As you can see, these yellow dots are sugars, so it is having plenty of sugar, so it's more glycoprotein than rest of the other immunoglobulins. Very very more glycoprotein structures. This is also present as monomers, and it is also having tail piece, as you can see here. Why? Because it is also need to interact with this B cell surface receptors in all these cases. Okay, IgD is primarily found on the B cell surfaces, where it functions as a receptor for antigen, because we know that this B cells can also act as antigen presenting cells, or APCs. So it must have something to recognize antigen, to hold on to antigen and process them, right? So for that reason, because antigen presenting cells always work like that, they hold on to antigen, they'll process them internally, and they will again uh, hold it outside, so that they can put this information throughout the immune system. So this kind of a receptor is made by this kind of IgD molecules that we can find here. Okay, IgD is not having any capability of binding with complement, and it cannot fix complement. Now IgE is another. IgE is also the latest common serum immunoglobulin, and it is much more related to a kind of hypersensitivity type 1